today I will pursue some very interactive points like genes and evolution. Okay. What is genes and evolution, and what is level of organization? What is cell, cytoplasm, and organelles? What is diversity among organisms? First of all, what is genes and evolution? Of course, I have already given you a lot of things between genes and evolution. Particularly, our cell is just like a very diverse world. If you see, diverse world means it can be, if you see the history of the cellular organization. What are the cells? Basically, one question immediately arises among themselves. What are the cells? What is cellular organization? And what in fact the level of organization? So this is the $1 million question. How is the cell is originated in the life, and how the life exists in the cellular organization? What is the interaction between the cellular organizations and gene? Cellular organization and the gene. What is the interaction between the gene and cellular organization? If you see the cytoplasm, and organelles, the cell is just like the compartment, okay? Like this compartment, in which you can see the different things. Like you can see the air conditioners, you can see the tube lights, you can see the fan, you can see the all the things. What it is like just a chamber, compartment system, cell, isn't it? Question arises here: how a nucleus is the master of the cell, because it's controls all the activity of the cell. All the activity means like just the metabolism, just like the maintaining the identity of the cell and maintaining the whole genome. If you see on the biotechnological field, because several times I have told you these things because you belongs to the technical background. Okay, what is the utilization of the techniques basically? All of the things you will study in these levels in the BSc honors like other degrees, but you have to emphasize the techniques. You have to emphasize the different patterns in which you are able to visualize the cell organelles. This is the main application of techniques. This is the main application of the degree. By means of which there are so many techniques. The first of all, if you hopefully you remember lastly what I told you. What is the backbone of the biotechnology? I will remember DNA. How the DNA? If you say the DNA is the backbone of the biotechnology, immediately one question arises: how will be the DNA? How will be the structure of the DNA? At least what is the orientation of these DNA inside the cell? And these, what is the interactions between the DNA and the proteins? If you see the life history of the cell, if you see the life history of any cell, like prokaryotes, eukaryotes, and you can see the life history of plants, you can see the life history of animals. What is the initiation process begins from that? That is cell differentiation, isn't it? Cell differentiation. वो एक point, वो जीवन का अहम हिस्सा जिसमें जीवन की शुरुआत होती है. That is the big interaction between the genes and evolution. So the most important parameter of these techniques, basically you are going to study genes and evolution. That is how the life originated, how these cells appears in each one, how a single cell form, either in prokaryotes, either in eukaryotes. This is the genesis. You have to understand that. How the genesis of the cell it originated. So that's why now question arises here. That's why we have added this word evolution on this. You must know, if you are talking about the cellular organization, you are talking about the compartment system of the cell. It was in begin fact what was the origins? क्या जो हमारे ancestors हमारे ancestors थे, जहाँ से लाइफ की शुरुआत हुई. What was the root? How the single cell arises if you go back? If you go billion years back, this is the $1 million question. You have to understand. So this is the big interaction. This is the big interaction between the cell and gene. So basically, I just want today the fundamental interaction between the genes and evolution. हालांकि so many times we always talk about these things, genes and evolution, but today we will interact in able to visually. So that's why I just want to more specifically to tell you these things. What are the utilization of the gene? How a gene is formed? One question. If you know what is gene, okay, this is the hereditary unit of the gene. Likewise. These are the different definitions of the different if you define the gene and how is gene is oriented in the cell. In the second question, how these cells is oriented in the cell, what is cellular machinery and what is the organization of genome. Organization of genome in different cells. Like if you're talking about the bacteria, that means you're talking about the prokaryotes. If you're talking about the eukaryotes, so there's a diverse living world of the eukaryotes. That means if you're talking about the fungal cells, you are talking about the protozoan cells, you are talking about different rest cells you may include in this diversity of the cell. The first things you have to understand the basic title, basic utilization. If you're going to study this title of this semester, you must know about what is the real utilization of this level, this title you are going to study. I think you understand? Gene cell evolution. Okay. And then how is these three terms interact with each other? Why the evolution we have added this word with the gene and the cell? This is again you have to understand. And if you remember, I just told you several times 5 levels. And the first initiation point, that means first initiation point, the first initiation level of organization where life begins, that is chemical level of organization. There is the assembly. But that means this is the just you can say that without chemicals there is no any assembly in the circulation of the cell. Because the first thing every biomolecule is made up of chemicals. Every biomolecule is made up of biological molecules, and these biological molecules plays a very significant role in the whole vital biological process again in the different cells, like eukaryotic cells and prokaryotic cells. Because before these level of organization, this is the fundamental things you must know between the difference between the prokaryotic cells and the eukaryotic cells. Because if you see the diversity among organisms, so there is a two types of diversity among organisms. Note it very carefully. Diversity among organisms, that is one thing. Diversity may include, you can see the simple example of the rhizosphere and the rhizosphere. If you see the nature, there is the huge diversity of the nature. Like you will able to interact different microorganisms in the rhizosphere. You study so many plants and the fungal cells. They are integrated antagonistically. Either they may be parasitic, either they may be saprophytic, either may be independent, either independent to each other. So there is a two criteria of diversity of cells. You must always remember. Diversity among organisms, one thing, and diversity among different shape and size. Is that clear? Shape and size. If you go intracellularly, any cells. If you go through any cell intracellularly, that means if you going to study the intracellular levels में उसके सेल के अंदर के आपको स्ट्रक्चर जब विजुलाइज कर रहे हैं, उसके अंदर उसकी डाइवर्सिटी क्या है, shape and size. So you can visualize the smallest cell size. You can see the different. There's a huge diversity in the shape of the cells, likewise all the things. So again, cytoplasm and organism. This is also part of the cell. If you're talking about the eukaryotic cells, so you have to be attracted between the different cell organelles. Likewise, I started from nucleus. Like this is also called in the master organelles of the cells. If you go through again significant, that plays again significant role. If you go through the eukaryotic cells, like that is mitochondria. If you go through mitochondria, you can say this is the energy currency of the cells. Because if you're talking about the eukaryotic cells, without mitochondria, there is no any. If you have to any expenditure of the ATP molecules which are going to govern into the cells, you can see they provide the nutrition. Likewise, these whole data, they have to be governed by the different second master organs of the. If you're talking about the eukaryotic cells, that is mitochondria. That's why it is also called the energy currency of the cell. Okay? So they have different roles. I have just instance of this room because this is also this room is the closed chamber. Well, this is the closed chamber. Likewise, the all things are there. They have different roles of the cell. Likewise, cell is also the compartment. Okay, cell is different compartment. This is the entry point of the cell. This is the different cell. They have to be interacted with each other. That means these are the neighboring cells. By which there is some process in which they have to interact themselves. Interact themselves means like this the neighbor. Side by side, there may be exchange of solvent particles, any ions, different molecules. They and then the interaction between the two different cells. So, if you're talking about the plant cells, isn't it eukaryotic, prokaryotic cells? Is these interactions occurs in prokaryotic cells? Because these are the unicellular cells. And the one again questions diversity and then cellular organization, and the again one the more important, how you can isolate a single cell. Again, this is a very important question for you. If you see, if you understand all the things, if you understand the diversity in the organisms, if you understand the cellular organization, and if you understand the significant role of the biological molecules, and thus this is very important also. That means how you can isolate the cell in the organisms. I'm again coming on that point. Again, a significant role of biological interaction. That is there. I just come for the seventh point. Interaction between the biological molecules and the cellular organization. If you study all different levels of organization, if you study all things, that is very important thing that how a biological molecules interact and how a biological molecule is responsible for the cellular organization. Previously we talked about the DNA, and then that DNA converts into its RNA. Again, there is a very complex process. How its DNA turns down to the RNA, and these RNA turns down to its own proteins because they contain its identical information, genetic information in coded and encoded language, in which it will be able to express in the next generation, which is totally responsible for expression of the traits they are just found in their parents' cells. So this is the important thing, that how these biological molecules is responsible and how these biological molecules are interacted with the formation of the cellular organization. And metabolism, anabolism, signal transaction, cell communication, cell death, cell synthesis. This is the whole significant role. If you're talking about the very use and the use word, you can see the biological process. There are so many biological processes coming on inside the cells. If you're talking about eukaryotic cells, different cells, prokaryotic cells, these are the different cells. Because if the biological process is going on, and then I have again discussed with you. Next, I have not mentioned like what, but I was able to this information right to. And the next, after the significant role of the biological process, that is interaction among organisms. Interactions, what is the interactions among organisms? You can say, if you are going to study the interactions between among organisms, this is just not the interactions among themselves with a person like this and this. This is the biological interaction between organisms. If you see, several times I told you, if you go through the nature, so there are the different types of microorganisms out there. Some are visible, some are not visible, some. That means we are talking the visible microorganisms we are talking about. That is, we are talking about some fungal cells. There are some because they come to the category of microorganisms, but they are visible. But most of the microorganisms are not visible. So you must know how the interaction in the soil between the microorganisms and different living world, plant cells, animal cells, protozoans are there. What are the antagonistic effect? What is the saprophytic effect? What is the parasitic effect? So you must know that if you go through the cell. Just for instance, suppose I just convey you that you have to isolate a community of bacterial cells in a particular soil sample. Maybe soil sample may be any concerned to the forest or in the field. What you will do? First of all, you will prepare a soil sample, okay? And then what will you do? You will do a dilution rate method. Sample, in that sample, you have to go through the different dilution rate method, and then you will able to derive a single cell. If you are talking about the prokaryotic cells, and if you are going to derive a different cell like eukaryotic cells, and you have to perform the different criteria of parameters. So you must know what are the interaction between the organisms of living world. If you are talking about the prokaryotes, what is the parameter of isolation of these cells in the laboratories? This is the you must know. So these are the different parameters, and these are different protocols. Microorganisms, either any interact each other in the nature. And then one again the questions arises here, that is how these survive for the betterment. How the cell is survive for the betterment, and you can able to law of determinism and the Darwinism, you see what is the principles in the nature, survival for the fittest. If the cell is going to survive betterment, if there is able to get the extreme environment, if any cell is getting any extreme environment, then it will survive. If any cell is not getting the extreme environment, it will either it can die. So extreme environment is the most important parameter in which a cell is going to survive. And finally, if you go through these all interactions, whatever we have to share with you, finally, if you go through the gene, because we have to start with the gene, and this is my wish to finish my lecture on the gene, because so many things we discuss among themselves. And finally, if you go through all the biological process, all the cellular organizations, all the cell cytoplasm, organelles, level of organizations, and finally the things begins from that, and finally start with that significant role of the nucleic acid and proteins in the maintaining cellular organization. That means transcription and the transmission. The process in which DNA converts into the RNA, the process is called transcription, and the process in which RNA is converts to its protein. This is the complex process, and this is the linear evolution of the life. If you study these linear evolution of the life, then you have no problem to study the genes and evolution, because you must know about the linear evolution of the life. Because every genome has its significant role in region. Because this, you can see the diversity among themselves. There is all due to the polymers of nucleotides. Polymers of polynucleotides. Pop means polymers of polynucleotides. That means we are talking about the proteins. And polymers of nucleotides, we are talking about the nucleic acids. Because every cell has its diversity, every cell has its own genome, and every genome has its own specific base pairs. That's why you can see that you are different among us. Nobody has identical members, right? Except some twins. If you see these things, that you can see every genome has its own specific base pair in the cells to maintaining the diversity among themselves. And finally, if you go through the universal truth of the cell, that is cell senescence, when biological process goes across the life, and they have to perform differently. Every cell has its own life cycle. Every cell has its own life cycle. That is the basic truth. The cell, not only the cell, any organisms, any person, any living beings, कोई भी जिसे आप living world कहते हैं, they have to die. Because, and this is very important. It depends on the life cycle of the cell. Because every cell has to pass through its own life cycle. If you're talking about the prokaryotic cells, it has to go through its own particular life cycles. You can see bacterial cells go through its own life cycles when it performs the process. Hopefully, most of you know that the lag phase, log phase, exponential phase, and the death phase. Stationary phase, that is also called the stationary phase. The stationary phase and the exponential phase are the same. The exponential phase and the death phase, lag phase just going to start in the growth, and lag phase the complete growth occurs, and then in the stationary phase, that is the adult cell is there, and they are ready to evaluate the secondary metabolites. If you're talking about the prokaryotic cells, and if you go through the plant cells, lag phase. If you go through the plant cells, the process occurs in the nature that is called cell senescence, cell death. In the molecular process, in the biological process, this is called cell senescence. Cell death, cell senescence is almost the same term, but it differently used by the different parameters. Both in the cell communication and signal transduction, cell communication, and the correlation between the immune system. The most two important things left behind, how is cell communicating? That is very important. You have to understand also how is cell communicate differently. And what is the significant role of this communication in organization of the whole system? In the organization, the whole genome. If you see immune system, immune level of the organizations, there are some partitions, there the distribution among some cells, they have to perform differently. B cells, T cells, if you see on the volumes of cells, but they have to perform different cell duties. There is a particular system, peculiar system of the cell organizations, which occurs differently. If you are talking about the prokaryotic cells and the eukaryotic cells.